Nervous system, part 1. The nervous system is the most complex system in the body. The brain serves complex functional, regulatory, and integrative roles that underlie the functions that maintain the body, as well as provide the identity, personality, and relational capacity. The brain also exhibits plasticity, which is the ability to adapt to altered internal or external signals in ways that might be either positive or negative for the functional status. The nervous system regulates general homeostasis, both through the autonomic nervous system, which is the parasympathetic and the sympathetic nervous systems, as well as by neuroendocrine control via the hypothalamic pituitary target gland axes. Diseases of the brain and brain trauma can cause deficits ranging from the loss of perception, strength, and coordination to permanent vegetative states, as well as alterations of personality and lost or diminished social abilities. The brain consists of neurons and glial cells, along with supporting structures and blood vessels, with an outer protective membrane coat consisting of meninges. The brain connects to peripheral tissues, which are skin, muscles, joints, internal organs, and glands, through the peripheral nervous system. The information enters the spinal cord and the brain from the periphery through the sensory or afferent axons. The information from the spinal cord and the brain is carried to the peripheral muscles, organs, glands, and blood vessels through the somatic or autonomic efferent motor fibers. The brain and spinal cord are also surrounded by 3 meningeal layers, the dura, arachnoid, and piedmont. Between the arachnoid and the piedmont is the cerebral spinal fluid that further will cushion the brain and reduces brain injury after a trauma. Cerebral spinal fluid also circulates internally in the ventricles of the brain, which are 2 lateral ventricles and the 3rd and 4th ventricles. Additional protection from circulating systemic toxins is provided by the blood-brain barrier. These are additional nervous system disorders. The surface of the cerebral cortex is made up of gray matter, which are neuron cell bodies that are organized in layers. The forebrain consists of cerebral cortex and deeply embedded white matter, which is made up of axons, the basal ganglia, the thalamus, the hypothalamus, the hippocampus, as well as the amygdala. The forebrain connects to the brain stem, which consists of the midbrain, pons, and the medulla. The corpus callosum, a white, thick matter bundle, connects the right hemisphere to the left hemisphere. The basal ganglia, the nucleus accumbens, and the thalamus are buried deep within the forebrain. The limbic system is a general term for several structures that have been implicated in emotion perception and regulation. The structural components of a neuron are the cell body or the soma, the dendrites, axon, and axon terminal. The cell the cell body of the neuron contains much of the cytoplasm and many of the organelles. The organelles include the nucleus, endoplasmic reticulum, Golgi apparatus, ribosomes, and mitochondria. The dendrites have small projections that are termed dendritic spines, which are the site of many synaptic inputs. Each neuron also has a single axon, which is an outway path that branches as it projects to the target regions. At the end of each axon branch is the axon terminal. The axon terminal contains synaptic vesicles filled with neurotransmitters. Neuronal communication is both electrical and chemical, beginning with action potentials generated at the cell body and propagated to axon terminals, where then the chemical transmitters are released. The distribution of characteristic membrane proteins to specific portions of the neuronal membrane is responsible for the functional properties of each part of the neuron. The sodium-potassium pump is found in the membrane of every part of the neuron, and it is responsible for generating the differential distribution of ions that contribute to the resting membrane potential. The presence of potassium leak channels gives neuronal membranes their highest permeability to potassium while they are at rest. Therefore, the resting membrane potential is close to the potassium equilibrium potential. The 3 voltage gated ion channels labeled at the lower right noted at the lower right shape most of the neuronal functions. Fast sodium channels are responsible for the upstroke of the action potential. The great density of sodium channels at the axon cause the action potential to be generated here for most of the neurons. Delayed potassium channels open as the sodium channels are inactivating. These 2 events, which are the delayed potassium channel opening with the sodium channel inactivation, lead to the repolarization of the membrane back toward the resting membrane potential. Slow calcium channels are found at axon terminals. When the axon potential arrives at the axon terminals, the opening of calcium channels allow calcium entry that directly leads to exocytosis of vesicles of the neurotransmitter. The resting membrane potential is produced by the differing ionic compositions of intracellular and extracellular fluids, the selective permeability of the neuron membrane when it is at rest is more permeable to potassium than any other ion, and the sodium-potassium pump, which maintains the ionic composition of the intracellular and extracellular fluid. The potassium moves out of the cell through the potassium leak channels until the inside becomes negative enough to counteract the force of the concentration gradient that favors this outward movement. The negative resting membrane potential is necessary for the neuron to respond to stimuli and conduct impulses. If the membrane is sufficiently depolarized by synaptic or sensory inputs, a threshold voltage will be reached where the fast sodium channels open to initiate the action potential. The sodium channel opening allows sodium ions to rapidly enter the cell, down their concentration gradient. At the peak of this action potential upstroke, the sodium channels close and are briefly inactivated. At the same time, delayed voltage-gated potassium channels have reached their fully open state. Potassium flows out, repolarizing and eventually reestablishing the negative resting membrane potential. A synapse is a point of contact between the presynaptic terminal of one neuron's axon and a postsynaptic membrane of the target neuron. The axon terminal is a swelling at the end of the axon containing small transmitter-filled synaptic vesicles and mitochondria. The terminal is separated from the postsynaptic membrane by a narrow synaptic cleft. The sodium-potassium pump on the membrane is to maintain the proper ion gradients and the membrane potential. The voltage-gated calcium channels that open when the action potential reaches the terminal. The calcium entry signals for the release of transmitter vesicles by exotosis into the synaptic cleft. The secondary active transport membrane for the uptake of the transmitter into the terminal prior to degradation or repackaging into vesicles. Neurons communicate by the process of synaptic transmission. If the inputs to a neuron provide sufficient excitation, that neuron will reach a threshold to have one or more action potentials. The action potential of a presynaptic neuron results in the release of a neurotransmitter. This is a chemical signal that excites or inhibits the postsynaptic target. If the postsynaptic neuron achieves sufficient excitation, it reaches the threshold for its own action potential, and the resulting chain of signals culminates in a change of state, behavior, or movement. To summarize, the changes in membrane potential created by the synaptic transmitters can either create excitation, which favors the action potential generation, or they can inhibit, meaning inhibiting the action potential generation. Two major classes of transmitter receptors are found on neuronal membranes: ionotropic receptors and metabotropic receptors. Ionotropic receptors are also known as ligated gated ion channels. They are multimeric proteins surrounding a central core, typical of ion channels, and they are selective in their ion permeability. Metabotropic receptors are G protein coupled receptors that create slower changes in membrane potential through second messenger cascades and indirect opening or closing of ion channels. Signaling by metabotropic neurotransmitter receptors can be thought of as a slow modulation of ongoing activity.